Good evening, everyone, and welcome to the Real Science Exchange, the pubcast where leading scientists and industry professionals meet over a few drinks to discuss the latest ideas and trends in dairy nutrition. Hi, I'm Scott Sorrell. I'm going to be your host here this afternoon. We're at the Real Science Symposium. That's a Balchem part of our Real Science media. We have symposia, we have podcasts, we have webinars, and we recently introduced something called the Real Producer Exchange, where we interview dairy farmers from around the world. And it's an opportunity for other dairy farmers to listen in on best practices, what worked, and even what didn't work. And so that's a section that we focus on quite a bit as well. But this is the Real Science Symposia, where we do these symposia around the world and in person. This Real Science Symposia was titled Redefining Amino Acid Input, New Research Unveiled. So we had several speakers today, the first of which is Dr. Mike Van Amberg. And Mike gave us two presentations today. One was kind of technical in orientation, and it was titled Amino Acid Nutrition for Increased Milk and Milk Components. And so the second one he gave was more applied, and that one was titled Feeding Cows for 1 ton of components and 2,000 kilograms of milk per year, right? So that was meant to be a little aspirational. 20,000, what did I say? 2,000, it's 20,000. There we go. All right, Mike, I'm going to circle back to you. But to your right is Dr. Usman Arshad, and he's from the Federal Institute of Technology in Zurich. The title of his presentation was Lysine, a vital amino acid for maximizing milk production. And then finally, to my left here is Dr. Sean Richards. Sean's with Balchem, and he gave a presentation titled Driving Milk Component Yield with AminoSure. And so, then not to be left out, Dr. Stefano Vandani, he's going to be the co-host this afternoon. He's going to keep us in line here and ask some good questions for us. So with that, what I'd like to do, Mike, is start off with you. Just kind of give us a brief overview of yourself.
Well, I've been on the faculty at Cornell for 30 years this year. I advise, I still, as a faculty member, I'm kind of a full service guy. I still advise about 35 undergraduate advisees. I advise the Dairy Club. I lead the Dairy Fellows program, which is our experiential learning program, and I lead the CNCPS effort. So that's kind of who I am. Every once in a while, I go home and I see my wife and kids.
Dr. Van Amberg, there he is. All right. Usman, this is not your first time to the pub. I believe the very first time I met you were actually a student at the University of Florida. So tell us a little bit about yourself.
Yeah, it's been 6 years working with Balchem, I would say. I am basically a veterinarian by training, and then I moved to States from Pakistan in 2018 to do my PhD at University of Florida with Jose Santos, working mainly on the choline and how choline can improve the performance in dairy cows. And then I graduated in 2023, moved to UW-Madison, working with Heather White. Did some trial there on lysine and amino acids and feed efficiency. And recently I moved to ETH Zurich, working as a research scientist and study director of the program. So I have 5 master's students at the moment and 2 PhD students where I'm co-supervising with Muti and you. So, yeah.
Excellent. Well, thank you for joining us. To my left here, Dr. Sean Richards. Sean, you've been with Balchem not all that long. Tell us a little bit about yourself and kind of your background.
Thanks, Scott. Yeah, I've been with Balchem almost a year now. Ruminant Technical Services Manager for Europe, Middle East and Africa. Before that, worked for a number of companies in the feed industry, come from a farming background in North Wales, and very passionate about dairy cows.
All right, good. And Stefano, you're going to be the co-host today and color commentator. So you're a bit of a retread, right? You started out with Balchem, you left, now you're back again.
Yep.
How long have you been gone? Tell us about yourself.
So, well, yeah, this is my second experience in Balchem. I've been with Balchem 6 years, then I left, and now I'm back since April. As a background, I'm a beef guy, actually. So I have a PhD in beef cattle nutrition and management. As I always say, I turn to the black side of the ruminant for work, for my job. So, but yeah, I'm passionate about ruminant, I would say. So I still work on some time on beef, but majority of my time is on dairy.
Well, welcome back. We're glad to have you.
Thank you.
Mike, going to turn to you. Give us a brief overview of that very first presentation you gave. It was technical and oriented, but let's start with that one.
Sure, Scott. I think a short version of that would be that we spent a lot of time trying to figure out, as we were building version 7, how to integrate energy and protein to represent, the requirements of a lactating dairy cow. We stumbled on an approach where we could integrate amino acids directly with energy, and then we started testing it. And what we found is that when we gave them more amino acids, they actually responded. And sometimes they responded in ways that we didn't fully appreciate, like, why did amino acids increase fat synthesis? But that led to some, relearning, how the mammary gland does partition nutrients, and that helps us figure out how to improve milk composition and also energetic efficiency.
Yeah. Mike, one of the slides that you've given several times that I'm kind of enamored with is the one that shows the genetic improvement over time. And like the last 5 years, actually it's probably 10, but it's been changing exponentially. So my question is, you're talking a little bit about requirements and trying to meet the cow's requirements. Do we really know what they are today? And then where are they going to be in 5 years from now? And how do we keep up with that?
That's the question that's motivating my research team right now. We've got some nuts and bolts things that we're working on, but some of the visioning stuff is how do we set up studies to actually test that, Scott? That, this sounds a little bit silly, but I would say I don't know if we really know their requirements.
Right.
I really don't think we do. Right. And there's some, what I didn't talk about, right, just to that question. One of those herds that I highlighted, that 4,000 cow herd in the north, it's got that barn or that pen full of those high genomic cows, those Holsteins. When I called him a few weeks ago, I was talking about wanting to do this genotype by nutrition interaction study with them. And after a little while, he goes, "Oh, that's what you want to do? We're already kind of doing that." And I said, "What do you mean?" He says, "Well, my nutritionist and my genetics counselor were here, and they said, if you've got that kind of capacity, why don't we play around a little bit?" And I said, "Well, what did you do?" He says, "We quadrupled the amount of those amino acids you tell us we're supposed to feed."
Yeah.
And I said, "Really?" I'm driving down the road at, 75 miles an hour, and I'm thinking, I don't know if I should pull over or if I should just keep driving, because my brain went someplace else, right? I wasn't focused on the road. And I said, "Okay, so tell me what happened." He goes, "They improved their productivity. And that's how they got to 7% fat in some of those cows." And I said, "Okay." Interesting. It's fascinating, right?
It is. And I got so many questions kind of around this kind of whole area. I don't want to pick on you, Sean, but during your presentation, you said you were showing us what % of the methionine and lysine were being met. And the thought occurred to me is, by what measure? By what requirements? And are we even farther underwater than we think we are today? And then just to build on that, right? I mean, Mike then did
I like your word usage, underwater, right? I don't think we're underwater. We're just not optimizing. How's that?
All right, fair enough. The other idea you had is comparing the Fiat to the Ferrari. And then I was asking myself, do we have a barn full of Ferraris and just treating them like Fiats?
I think so.
Yeah.
Yeah. No, I don't think it's that bad.
Okay.
But I think that's a good analogy of what the potential is if we could figure out how to tell them to do it.
Yeah. All right. All right. Scott's going to quit talking now. Stefano, got some questions from the audience?
Yeah, I mean, there's multiple questions about the UK being a liquid milk market mainly. So one of the questions was, would you change the approach to amino acid balancing in a liquid milk, not pricing protein and fat? And the other question was, what are the other benefits that you can get out of amino acid except what you present today, that was milk production, protein, fat content, and so on?
Is that for me?
Yep.
Okay. So that's a common question. What if my milk market doesn't pay for components and I just want to get more volume? So what I didn't show today is a study where we asked some of those same questions, but we started in the close-up period. and we fed a control diet in the close-up period, typical close-up dry cow system with whatever, 1,100 grams, probably around 1,100 grams of metabolizable protein. And then we had a treatment where we moved that up to 1,600 grams of metabolizable protein. And then that was in the close-up. And then in the fresh period, we did the same thing where we had a typical fresh diet, and then we enhanced the MP supply in the treatment. I can pull up that data sometime and show people. I'm not going to remember the grams per mcal right now, but there was a significant difference, like 400 to 600 grams difference in MP supply. Altogether, from the control to the high was about 7 kilos of energy-corrected milk. All right? And it wasn't components, it was lactose, right? So I think to your question, in a fluid milk market where you're not being paid on components, you got to start at the initiation of lactation, right? So start it prior to calving, get your MP up. The moment they calve in that fresh pen, you've got to push your MP up, and then you've got to reinforce that, because if you're going to get, 5 or 6 or 7 kilos of milk up to peak, you've got to reinforce that all the way through, right? And but that's a lot of milk, and that's a lot of milk volume. So that's, I think, I think that's how I would tell people in a fluid milk market how you're going to make that work. The other things that we know of, and this is such a hard thing because the problem with asking about what about all the things like immune system, inflammation, repro, all those things, there's only a couple of repro studies where there's actually been longitudinal data, right? And Sean talked about that. When it gets to the immune system, we know there's a huge demand for amino acids in immune system responses. So I think we probably enhanced the immune system response, but again, nobody's done the challenge studies directly to figure that out yet, right? If there's a hole in our research, there's one where we do challenge studies at good amino acid balancing and not.
Okay.
Great answer, Mike. If I can just come in and add to that, I think very much here in Europe, here in the UK, you see that lysine and methionine are both closely limiting. And I think one of the challenges is if people are looking for components, you can add methionine and see a milk protein response. But if you want to drive yield, then you've got to true amino acid balance. And I think that's where the opportunity comes. I think lysine, we don't have blood meal in Europe, so I think our lysine levels are consistently lower than US type diets. And that is where the opportunity comes to be able to lift milk yield by using true amino acid balancing and bring that forward. And I think I'll add to the second question about health and fertility. I think, I had a slide in my presentation that talked about a practical UK farm that saw, through amino acid balancing, I think 600 kilos more milk per cow per year, but actually felt the cost of the amino acids was paid for by improvements in health and fertility. And I remember going through the data with a consultant, and there were less cows with mastitis, there was less metritises, all the numbers moved in the right direction. And what was that a controlled trial? No, it wasn't. But to me, there's really strong evidence that if you meet the amino acid requirement, you help support the immune system and inflammation management that helps health and fertility.
Yeah, if Stefano allows me, I would like to add on what Sean just mentioned. We recently conducted an experiment at UW Medicine where we had a group of cows, 32 cows, where we had MP adequate diet, and then a group of cows, 32 again, where we had MP deficient diet. And the cows on the MP deficient, they were getting almost 250 grams less metabolizable protein whatsoever was required based on their production performance. And then what we did is we supplemented those two diets for 12 weeks, starting from the mid-lactation all the way to the end of mid-lactation. And then we collected the blood samples at the end of the 12th week, and then we isolated the T lymphocytes. And the T lymphocytes are very much necessary for the adaptive immune response. And once we isolated the T lymphocytes, then we run the seahorse analysis. We really wanted to see how the amino acid supply can alter the molecular and cellular pathways, not only just, oh, we saw less mastitis, we really wanted to see what's going on at the molecular level. So interestingly, what we observed is, so the flexibility to respond to a metabolic challenge of lymphocytes was reduced in MP deficient cows. In simpler words, so when we challenge those lymphocytes in the lab with or without some metabolic active agents, I don't want to go into detail, so the flexibility to respond towards that challenge of those lymphocytes was a lot more improved when they were having adequate amount of amino acids in the diet. So which tells me that immune system, they certainly need amino acids, and the function of the immune cells, specifically T lymphocytes, is enhanced when the diet is adequate in amino acids. This work was presented at ADSA, and the manuscript is under review JDS, so hopefully it will be interesting to read. Yeah, just wanted to add on that whatever you do at the farm level, it really goes into the cellular level, and body cows really respond to that.
Yeah, thank you, Usman. While you got the microphone, why don't you go ahead and share with us kind of a brief overview of the presentation you gave earlier today?
Yeah, so the presentation which I did today was based on the meta-analysis where we wanted to address a question because there is some uncertainty that should I supplement rumen-protected lysine, should I not supplement rumen-protected lysine. So what we did is we collected all of the literature whatsoever is available at the moment, and then we conducted a meta-analysis. And what we observed is among 12 experiments, 13 experiments with a little bit over 600 cows data. So we showed that cows which were supplemented with rumen-protected lysine, specifically the ones if you start in early lactation and the ones where you supplemented for a little bit longer duration, they produced 1.5 kilos more milk than those cows which were not fed by the rumen-protected lysine. So, which really tells me that supplementation of amino acids improves the productive performance in cows.
Tanya, I'd like to go to the audience, see if there's any questions out there. If you guys have questions, just raise your hand, and Tanya will get you a microphone. Anybody have a question for us? We got one over here.
Yeah, thanks, guys. Stephanie, you said I had to ask this out loud, not on the app. So I suppose mainly directed at Mike, but everyone else feel free to jump in. Given that there appears to be some flexibility of utilization within those group 2 amino acids, which obviously includes lysine, A, how do we account for this when rationing? And B, do we need to build it into ration formulation software? And I guess, how do we do that, if so?
That's a great question. I'll try to answer that as completely as I can to the point. I don't know, was this set up between you two or something?
Oh, no.
Okay. But if I back up a step, right, I talked about lobely, right? And I think the key to the whole thing is that we're probably not going to be able to describe the use of all those group 2s, right? But we know that they're going to go to non-essentials and they're going to be oxidized. And I showed you guys that the lysine and the histidine and some of them are going to go, into pathways to support enzyme activity and as carrier proteins for fat synthesis, right? So those are non-net uses, right? Which is why you see that uptake greater than the export. I think the way we calculated or estimated the requirements by tying it to energy and then finding an optimum in a lactating cow was probably the best way we know how to do that, because in that condition, the primary energy and nutrient utilization is to support lactation. But at the same time, all those other uses have to be accounted for, right? Which is why I made the point about, I think we make a gross oversimplification and mistake when we say, hey, well, I'm just, and the NASEM did this, right? You guys don't know the NASEM, but they did this where they drove all the requirements through milk protein. And that ignores all of those other uses, right? You can come up with an efficiency to do that, but it's really confounded by all the other things going around in the cow. So I think that's that global efficiency that we came up with as an optimum, not a maximum, right? Because the maximum would be more deficient, is the way we can account for some of that interaction. You know, to that point, you mentioned lysine. I can still hear Helene Lapierre in my head. We would have these, you know, this is quite a few years ago now, we would go to the meetings, whatever meeting, right, whether we're in Europe or we're in the US, and she'd say to me, Mike, why does the mammary gland take up all the lysine it sees, right, if it's such an essential amino acid, and not put it in the milk protein? And I would look at her and say, because it must be other metabolic uses, right? But all biochemical pathways will show that, right? And I didn't show that today, but, you know, lysine goes all sorts of places, you know, oxaloacetate, you know, we can go all sorts of different places there. So I think that the way we did that probably captures it for right now. We might figure out a better mousetrap eventually, but, you know, again, you got to look around and say, did anybody else do this? Well, poultry and swine operate that way, and it's worked for them for many years. So I'm hoping that as long as you can resolve the amino acids coming out of the rumen, then we should be good.
If I can just add to that, you know, I think great answer, Mike. If you look at a typical Northern European diet, our lysine adequacy is somewhere around 85%. Now, you know, are we balancing to get to 100%? Yes or no? But we are getting, if you improve that number, you'll see a response. So are you going to 100 or are you going to 97? But you will see a response. And that's how you build it into practical ration situations, is you build it in as a trend in the right direction. And, you know, we may, you know, when Mike reoptimizes his amino acid requirements and we come up with the next generation, you know, the numbers may change, but we're making a step in the right direction. And I think one of the key messages I've got out of today, and I think Mike's used it a number of times, is improving the energy efficiency of a cow by amino acid balancing. And that is absolutely key to how we balance and ration cows.
Yeah, and I can add, I did the same exercise that Sean showed today in his presentation for Italian diet and Middle East diet. It's exactly the same. Wherever you don't use a source of lysine like blood meal, you have a lack of lysine, you have a lack of methionine as well. So I would say it's a very common problem for all the dairy rations. So it's something that we need to improve, and complete balancing of amino acids is where we have to go, the direction that we have to follow.
Thank you. While we're on the lactose subject, in my innocent way of looking at it, lactose is the osmotic driver to drag milk volume, and lactose directly reflects blood glucose. We use it as a default measure. But what you're saying is that simple serial transition of glucose to lactose through the udder is actually much more complicated than we're led to believe. So I'm not sure if you can explain federal milk orders to us in the time we've available, but we have an even more complicated pricing structure for milk over here, with every dairy having variations between white water and paying for solids. So more often than not, we have a challenge of people who are producing unnecessarily high solids and would actually get more income by having more volume at a marginally lower solids, than we have people who are struggling for solids percent actually. So with your explanation now of the relationship between lysine and lactose, you may be revealing a different approach for us on those situations where we can get more income by having more volume, less solids.
So this is I'm going to give a very complicated answer, because it does come down to milk composition. So one of the things that we're noticing is as we change nutrient supplies and signaling, that we can redirect the carbon within the cell. And it's a fascinating thing, right? Because it's driving Barbeno crazy, because I keep asking him questions at the root of, he goes, we got to look at the ribonuclease activity. Okay, Well, but we're not going to do that routinely, all right? So how can we do it? Well, we can do freezing point depression on milk, because here's where that goes. So as we get, and this all comes back to trying to figure out how to turn the protein synthesis on. So one of the things that we've noticed is when we do this, the milk fat will increase, the milk protein sometimes increases. When that happens, the lactose as a proportion decreases. Now, if you put it on a fat-free basis, it comes back close to normal, but not fully. So my question to Dave a few weeks ago, we both work too hard. It's a Saturday or a Sunday, and I'm sending him emails, and he's responding, and he's a little upset with me because I'm challenging what I think we know about milk composition. But here's where I'm going. Because one of the things that a heifer does, and this is fairly well known, a heifer never makes as much lactose, a first lactation animal never makes as much lactose as an adult cow because they still have a growth requirement, right? So they're putting that extra energy into the growth requirement, but they compensate by the whey component minerals. And that creates a fairly high osmotic pressure that offsets the lactose. So the really interesting thing is, as we get these cows that can make all this fat, they have the same capacity to make the protein. Because my question, to your point, my question to Barbeno has been, if we can turn this protein on and the relationship between casein and whey always remains the same, as we drive the casein synthesis up, we're going to bring that whey along, we're going to get those minerals, and there's less requirement for that lactose, which means we can get more volume by turning on the protein. And we know in body tissue, the water always follows the protein, right? So what I'm wondering is if we can turn that protein on, all of a sudden the milk volume is going to go up with it, and it's going to moderate the fat a little bit, right? And that's why that's why this whole thing about why the gland decides it's okay to make fat first and not the protein is really intriguing to me. I still think it's because we're short on leucine and isoleucine, right? There's just not enough resources to actually make it in most of these cows today. We've hit the next limitation, which is why you don't see it in the federal order data, right? It's not jumping at the same rate. So you ask a really good question. I just gave you guys a really complicated answer. But I think if we can figure out how to turn the protein on, then we're going to get that milk volume, and it's going to make everybody that question that was asked back there, that'll make that a lot easier to, how do we get into the fluid market? How do we get paid more?
My comment would be, do you think that is different for Mike, in terms of US to Europe because of, I would say, background, whether with blood meal, the US is nearer to amino acid requirement than Europe is. And do you think that changes that response in terms of lactose, or do you think it's the same?
The milk response is going to be the same. I think if no matter what you do there, if we can turn on the protein,
yep,
we're going to get. I think the volume is going to increase.
Yeah.
Right. I think the question is how do we tell the mammary gland it's okay to make protein, or how to get it to make the protein? I think it wants to, right? I don't think there's anything holding it back, except for the fact that it might not have all the nutrients that it needs. It's kind of like that conveyor belt thing, right? They're just not all there to allow it to do that.
How much, without looking at the mammary gland in isolation, how much is the interaction with general metabolism? I'm thinking things like, insulin, etc., having an impact on what, and perhaps mTOR, in terms of how the cow is prioritizing what it's going to do. Where does that all fit together, or do you think it is all at the level of the mammary gland?
No, there's definitely signaling involved. If you go back to the 90s, and I was tangentially involved in that because I helped make the diets. When Dale Bauman was trying to figure out milk composition and milk fat, he stumbled on some things by accident, kind of like we did, right? Dale was challenging the idea that insulin, if you got an increase in insulin, that would cause a drop in NEFAs. The drop in NEFAs because it's going to be held in the adipocyte. So now you're going to get less milk fat, right? You get milk fat depression by that. And that was an old idea. And what he found out so he would do these things called euglycemic insulinemic clamps, right? Where he'd infuse one heck of a lot of glucose in the jugular vein of a cow and then push the insulin up to keep the glucose, right around 100 or whatever number he was targeting in the cow. And what we found is that the cows made a lot more milk protein without changing the diet. So the moment the gland saw the insulin signaling, the first thing the gland did was make more protein. And it was significant. It's like 18 to 25% more protein. Right now that's short term because we didn't change the diet. Eventually they were going to run out, right? But it was very clear that insulin was signaling the gland to make more protein. So that's very important in this whole process. Now, there's arguments about that now in the current system, because I made a comment about that the other day at the Minnesota Nutrition Conference, and two of my colleagues, countered that and said, no, I don't think insulin's important at all. It actually shuts down protein synthesis. And I'm like, I don't think so, but I'm not going to argue with you in front of everybody, right? But that flies in the face of the clamp data, right? So I think, here's one way to think about this, and I said this to a group yesterday. So back to the milk volume question. We ran a study where we balanced all the amino acids, and the sponsor wanted us to feed chromium, right? And I like chromium through the transition. I wasn't really that much in favor of feeding chromium in established lactation. So we balanced the amino acids just like I showed you guys earlier. We had the lysine very, very high, about 250 grams. We had the methionine, the histidine, everything we're supposed to do. The cows are performing. With the chromium, they never budged on milk components. But again, I get the phone call from Barbeno, right? These are always, it sounds like a theme there, right? And it's almost like a comedy routine when you were there. And he said, what are you doing this time? And I said, why are you asking me that? He goes, you guys have the highest lactose I've ever measured in Holsteins. And I'm like, that's interesting, because my poor grad student was lamenting the fact that we didn't see the milk component shift, right? But we never dropped below 52 kilos of milk on average in that experiment. I mean, those cows made milk. We had cows hitting 70 and 80 kilos of milk in that study, right? Very high lactose. Now, they still made a lot of fat and protein because it didn't go away. But that, to the question about the insulin, so if the chromium is going to cause a clearance of the circulating insulin quicker, then there's less message going to the gland to upregulate protein synthesis. So it looks like the carbon defaults to the lactose and we get more volume. That's our interpretation of what we saw there, right? So that insulin signaling, I think, is very important. And that, back to the volume, if you're in a fluid milk market, that may be one strategy. We want to go back and test that one more time. But I went back and read all the literature, right? And there's a significant decrease in circulating insulin in every cow that was ever fed, beef or otherwise, it was fed chromium. So that would mean you're going to have less signaling at the mammary gland, so you could change components.
That fascinates me, Mike. As you know, early in my career, I worked on the impact of insulin on fatty acid synthetase and desaturase. And insulin increases milk fat synthesis and desaturation. And what you see when you feed lysine, you see that increase in fatty acid synthesis and probably desaturase activity. And therefore, you're having, in my view, somewhere an insulin or an insulin signaling effect. And if you can block that with chromium, you may drive more milk. It's going to be very interesting.
That's our next study, right? We're going to do a study where we're going to do amino acids and fatty acids at the boundaries of what we know, with and without insulin, and with and without chromium.
That would be a fascinating study.
But we're going to do biopsies at the mammary gland and the liver, right? So we're going to work on those mechanisms, because it's SCD insulin sensitivity is not described in the mammary gland. So if it's conserved like I think it should be, then it should parallel what you found.
Yep.
Yeah.
Yeah, that would be fascinating. I will wait to see those results.
So Mike, you gave a second presentation that was more practical oriented. Can you kind of give us an overview of that? What were some of the key takeaways there?
I think I forgot it already. No, I'm just kidding. You guys have made me, that whole milk composition thing gets my brain in another spot. What did it?
Podcast.
Oh, yeah, the 20,000 kilo cow. I think the point of that, Scott, is we have, you know, if you think of cows as athletes, we have a lot of athletes in our barns. We have tremendous potential. I think we have to think differently. You know, there's always the very practical things about cow comfort, cleanliness, water availability, lying time, all of that. But that, you know, as the cow increases her capacity to make milk, that stuff becomes much more important and more relevant. But I also think because we're building these animals that have such capacity that, you know, very early in life, we have to treat them like those animals and reinforce that capacity, right? So the moment they hit the ground, you gotta treat them like that animal that's going to break a world record. That's hard. That's a lot of management intensity. But when we do that, you know, and when we manage well, we see these cows responding in really good ways, right? And to the earlier point about repro and other things, you know, in those herds where we're doing a good job meeting all those things, those cows get pregnant, you know, they have normal dry periods, everything looks pretty good, and they last 3 or 4 lactations. High volumes of milk. So you just got to give them the need. You got to meet their needs, right? But you got to work a little harder to do it.
Yeah. So kind of related to that, maybe a little oddball question, but you know, you've often heard that you don't push your cows too hard. You're going to wear them out, right? And so I've heard Dr. Sanderson, I don't know if you remember Ken, but he was of the mind that no. These high-producing cows, if you give them the nutrition that they need, they're simply going to express their biology. And it's actually a symptom of health and not wearing them out. I don't know if you want to talk to that.
I talk about this all the time, more when I come to Europe, actually.
Okay.
Yeah, Scott, it is a pull system, right? The mammary gland is a pull system that's managed by communication between the mammary gland, the liver, and the brain, right? And the mammary gland wants to run as fast as it possibly can. That's the way it's designed. But if there's something in the environment that is not, you know, if they can't eat enough, if they can't lay down, if some of those things become limitations, then the brain and the liver are sending signals back to the mammary gland telling it to slow down, right? But if there's no limitations, and the cow can eat what she wants and lay down when she wants and has everything going for her, then there's not going to be that negative communication. So yeah, it's a pull. She's just going to go. And that is the best expression of the lack of stress that we have. Yeah, absolutely.
Good answer. Sean? Yep.
I think there's a question from the audience.
Sorry, again. The reference to athletes is interesting because human athletes tend to be reproductively compromised. It's essential in a productive dairy herd that reproductive ability is maintained, and you've all referenced apparent fertility effects on this. So high-producing cows which have successful lifetime careers are reproductively successful. So what have you got on reproductive ability with these very high yields?
So we are paying attention to that. Is there a direct effect of amino acids? Probably. There's not enough longitudinal data to actually look at that. Energy balance is important, right? Just like people who lose a bunch of body weight and don't cycle appropriately, that's going to have a problem. So I think it's just energy balance and things of that nature, but getting semen in them when they're in heat is really important, because I think that's the interesting thing, is that even when we see cows that are putting out high volumes of milk, as long as we can get them to cycle and we can put the semen in them at the right time, they still get pregnant, right? So it's not like they're anestrus. You just have to work a little harder. Now, I'm going to come back to my dairy farmer buddy who has those extraordinary cows that aren't just first lactation anymore. Now they're working their way into the herd, and he has a 30% preg rate. His jerseys had a 40% preg rate, and it always bugged him that the jerseys had such a higher preg rate, right? But I said the jerseys don't have the same capacity as your Holsteins, right? The Holsteins energetically are putting out a whole lot more than the jerseys ever thought of. So that probably creates more of a demand. But what he did say along the way is he said, I do think with some of these high genomic animals that we are seeing a little more lag in their ability to get pregnant. But what that probably means, and this comes back to a comment I made this morning, and then I'm going to be quiet, is that I think this is where we have to think about fat and fatty acids. There's very good literature about particular fatty acids that will stimulate, development in the ovary, function in the ovary, hormones, and everything that goes with it. So what that tells me is if we get these higher producing cows, we have to, again, keep thinking that fat's not just a bucket of energy, but that she has a particular fatty acid requirement. To do that, probably some of the longer chain essential fatty acids are going to be necessary in that cow at that point to be able to allow for normal ovarian function, and get that taken care of, right? So that just drives this idea of we're going to have to do a better job of thinking about more precisely her requirements on the fat side, just like we do on the amino acid side, to keep those functions going as well as they should.
And, if I can just add to that, I think one of the comments you've made several times, Mike, is by amino acid balancing, you see improved energetic efficiency. And I think that may be one of the subtle differences between your question, Neil, between the athlete of a cow and the athlete of a human, is that by getting amino acid balance right, you can improve the energetic efficiency. There are some nice studies out there that show, certainly in limited data, that things like methionine can increase lipid droplets in the developing embryos and giving more energy to that embryo to allow implantation. So, I don't think it's a simple answer, but I think energetic efficiency and some of the effects, direct or indirect, of amino acids on lipid droplets, on inflammation, have a positive impact on fertility and may negate some of the negative effects of driving milk yield, and it's that important balance.
Okay. I think we got a question back here.
Yeah, sorry guys, me again. Bit of a two-part again. Do you think that the response from amino acids in UK diets will be a touch lower given our considerably lower starch contents? And obviously that leads to a lower bypass starch level. Are we getting a much lower impact from that sort of amino acid sparing effect from a gluconeogenesis point of view? And are we sort of catabolizing a bit of that amino acid potential? I mean, Sean, you put up some classic UK diets there at 17 and 18% versus yours at 27 and 29, Mike. So what do you think the link is there?
There's no doubt that, anyone who's worked with me will know that I'm a believer in starch driving milk yield in UK diets, and thankfully we've seen a move up in average starch content in UK diets, over my career. There is an impact, but I think the key point would be there is an absolute requirement for those amino acids, and I don't believe we are limiting our response to amino acids by having too low a starch. I think we start from a lower base, generally in terms of amino acids, as I've said before, because we don't feed blood meal, and that gives opportunity for response. And, I will consistently say, that wherever I have used what I call true amino acid balancing, so lysine and methionine in combination, we see milk yield responses in the UK and, significant milk yield responses. Talking to someone at lunchtime today and, they were talking about where we rebalanced that and we got 3 to 4 kilos more milk. And so I don't believe the UK response will be less because of it, because we're starting from a lower base.
No, I agree. Yeah, starch, to me, starch is interesting. I don't think it's the driver, right? Yes, you're going to get more propionate. You might get more milk volume if you can get more starch in there. But I think as long as you stick to that concept that we talked about where, hey, if you're at, forgive me, I'm going to have to do this in mCals. But if your cows are intaking 60 mCals, I don't really think it's important whether that's partitioned between gluconogenic, acetogenic, or lipogenic type, substrates, because I think the way we develop that is independent of that partitioning. So they've got that requirement, that's their requirement, right? Sophie Lemaskay at INRA has challenged me several times. She ran a study in Jerseys where she infused starch, where she didn't really change the diet, and you would expect all the downstream things, right? You got a little more glucose, you got a little bit more insulin, you don't get a lot when you're infusing it, and the efficiency of use of the amino acids increased, right? But she infused the starch post-ruminally, so it was no different than Bauman's clamp. Because Bauman, if we go back to his clamps, we were feeding really terrible, terrible diets, dry diets, and we could still get a 25% increase in protein yield with the signaling from the insulin. And I think that's what she's showing. The problem is that if you take that same concept and you work backwards, if you fed the cow that same amount of starch, you're going to make propionate, you're then going to make some glucose, that's going to drive more insulin, but you also made a bunch of bacteria, right, that upregulates. So I think in the end, the efficiencies are the same, right, because if you fed it, you're just going to move the whole thing up. But we can create these artificial conditions where we short circuit some of the natural behavior, and we can see these things where it looks like, hey, you're getting something for nothing. So these efficiencies, because she would argue with me, you need to have variable efficiencies. And I said, no, I need an optimum, right? And I'm still going to stick to that. So I think, no matter what your diet makeup looks like, we just want to stick to those numbers until we get better numbers, and they're going to respond. I can't tell you how they're going to respond. I wish I could, and I definitely could retire. But if we could figure, but they're going to respond somehow.
Yeah, if I can add on, since I experienced 2 different production systems recently, last year I was at UW-Madison conducting a research at 64 dairy cows, and those cows in Wisconsin, they were producing 60 kilograms per day, and that was a research station, not a commercial dairy farm. So we did a study there where we had to supply blood meal to meet the needs of those cows in terms of amino acids, but the idea was to see the effect of rumen-protected lysine and methionine for some particular objectives. And then I moved from Wisconsin to Switzerland, where we have very high forage-based diet, and the starch was in Zurich experiments was close to 17, 18%, but we were feeding at UW-Madison close to 29% starch. So, but when I moved to University of Zurich, so we were conducting a study where we wanted to see rumen-protected histidine having low energy or high energy diets. So surprisingly, when we were feeding 29% starch in University of Wisconsin-Madison and versus 18% starch here at ETH Zurich, in both scenarios, the cows who were supplemented with additional amino acids, they produced a lot more milk. We're talking here 2.5, 2.6 kilograms more milk compared to the non-supplemented cows. So I agree with Mike and Sean that, yes, cows, if you provide them according to the requirements, irrespective of the starch content, I think they're going to respond.
So since you were talking about histidine, there are a couple of questions about histidine. And I leave to you who wants to answer. So what would be a good source of histidine right now, since there is not a real source of histidine? And the other one is, histidine is still an expensive amino acid. Would it be possible in future? What's your vision about that?
I really don't know about Mike probably would give you the insights of the application, but when it comes to the research, we have conducted Balchem, Camin, 2 Chinese companies, and everybody says we have room-protected histidine. It's just we cannot launch commercially. But we have tried one from China and we have tried one from Japan, and in both scenarios, room-protected histidine, it improved milk yield. So certainly, we really need to come up with a source of lysine which is cost-effective and also acceptable in European market as well as in U.S. market. So,
I think the challenge, and I think I totally agree with you, Usman. If you look at research papers, you will see rim-protected histidine from Balchem and a number of other suppliers. The technology exists. It comes down to, I would not claim to be a monogastric nutritionist, but there's not a great deal of histidine used in monogastric diets. So there's not a great deal of histidine produced in the world. And the histidine that is produced tends to end up in aquafeed. And that has a fairly significant cost. And therefore, when companies like ours look at the economic response to histidine, it is very marginal. But histidine is coming, and I'm fairly confident that, within the next year or two, there will be a AminoSure H or AminoSure XH, whatever the product is branded, in the global market. It's coming. When you hear Mike talk, as he did earlier, about the histidine requirement even being higher than equal to methionine, that immediately you start seeing the response. As Usman has commented, there are trials, there are meta-analyses out there that show positive responses in terms of milk yield from supplementing histidine. But and I'll come back to that from a European diet point of view. Histidine is very much third or fourth limiting. So, we've got to address first and second limiting before we start pushing on to third. But, to me, histidine will be the next commercial amino acid.
Yeah, just to build on what Sean said, right? It's all about price, and price is coming down. So I'm a little more bullish about the time frame, so all I'll say is stay tuned.
Yeah, I'm pushing, this is not specific to histidine, but I'm pushing every amino acid supplier out there right now to think differently. It's a lot of work to make a room-protected amino acid. But the production of them, right, part of the problem is we get a break on the lysine and methionine because the monogastric species use them a lot, so there's a lot on the market. Histidine is what, 16 cents a pound, 12 cents a pound, or lysine it's cheap, right? People are adding it to things just to increase the protein content because it's cheaper than protein. That's how inexpensive it is. What I would tell a bunch of these companies is we have to get to the point where we have commodity amino acids, right? And I've told a bunch of companies, if we work out the details here and I can show you that we would increase productivity of cows if they had to feed 20 to 40 grams of isoleucine and leucine every day, and you think about how many millions of cows would require that, and that's going to improve the milk protein, that's a pretty good slug of amino acids. Somebody should get into the amino acid production game alongside the protection thing, right? I mean, I hear companies saying, I want to innovate, I want to innovate, I want to innovate. Okay, the innovation here would be get the amino acid production line and then go ahead and also protect it. So now you're vertically integrated and you can own that market until somebody catches up to you. You guys hear that? Okay.
That's my idea.
But I think I think the other thing about this though is we have to get to the point where forgive me people in the back in the table there, Kim, the marketing people have to relent a little bit, and because it needs to get to, instead of these high level branded products that have such a premium sometimes, right? We've got to get to the point where they're a little more commoditized so we can get it into more mouths, right? I think it's one of those things where, we got to decide where your margin's at. Is your margin going to be on volume, or if it's going to be on what it is right now, right? So that's I know that's a different topic, and it's been a long time since I've been on the other side of the table, but I was there once, and I understand that process. So, yeah, we have to think differently.
Message received, Mike. We'll pass it on to Balchem management. If Usman, if I can just ask you a question, I think your data was really good on the impact of lysine. Now, I think I was right in saying that the response of lysine, of feeding ring-protected lysine, took 70 days in mid-lactation. It was sooner if you fed it in early lactation. Any thoughts on why the time period, why it took 70 days?
Yeah, I mean, from the analysis point of view, we chose to be in the middle point of the data. So whatever data we had, 70 days was the median point. So we had to cut off at some point. So we decided to cut off in the middle. But from the lactation stage point of view, I guess my understanding is in the early lactation, I mean, where cows are gradually increasing their dry matter intake, they're also losing lots of amino acids. So just think about, she just give birth to a young one, and now she had this much uterus size, and over the next 21 or 30 days, she has to go under to the uterine involution, where she's going to lose massive amount of amino acids because she's going to lose lots of uterine tissue in terms of lochia and all other things. And also in early lactation, cows are going to lose body weight. They're going to undergo proteolysis. So that's probably one of the reasons that in early lactation, cows respond much more to the supplementation of amino acids, and probably that's the time when cows are in the need of much more balanced ration in terms of amino acids. But when it gets to the mid-lactation, yes, they also start improving their body condition score. On the same side, they are consuming much more balanced and steady and consistent dry matter intake. So I think, yes, that's why we observed a more magnitude in terms of milk yield in early lactation compared to the mid-lactation.
Do you think some of it is to do with amino acid, if I can use the term amino acid storage in terms of muscle mass, and it takes a while to replete stores if you feed it later in lactation?
Yeah, I would say that specifically histidine. So histidine can come from the muscles. It is stored there. So yes, it's possible that in the mid-lactation that cows can utilize their stored amino acids to partition towards the milk synthesis. But in early lactation, I believe that's the time when cows are deficient, not only in energy, but also in vitamins, nutrients, micronutrients. Among them, amino acids is the major chunk, I would say.
Yeah. Sean, you were one of our featured speakers. I want to, as we get ready to close here, give you an opportunity to kind of give us an overview of what you discussed today in your presentation.
My presentation today was to very much about putting the practical ration situation, putting the science into practice and putting some real UK, Northern European diets showing where there was limiting amino acids and showing that in, all the diets I showed, I think I showed 100% grass silage, 50-50 grass maize, and 30-70 grass maize. And in all of those diets, real UK and European diets, methionine was first limiting, but closely followed by lysine. And histidine, I think, was third limiting in 2 of those 3 diets. And it shows the opportunity. But I would say, typically those diets were about 75% requirement for methionine, 82-83% supply limited for lysine, and then histidine was in the 90%. So it shows the potential response of balance, of true amino acid balancing, of lifting all amino acid supply in terms of response. And I show practical numbers where we have trial data, where we have increased milk yield and milk solid yield through amino acid balancing and shown economic responses. And then I talked about the role of the Balchem AminoSure products in terms of consistent bioavailability and TMR stable products, and talked about how, through using science for methionine, the seleno-methionine method, to demonstrate good bioavailability, and also talked about lysine and particularly the new AminoSure XL that we will bring to Europe very soon, and the bioavailability of that and the data behind that. And it is all about how to use amino acids practically to deliver economic returns for European dairy farmers. And that's key. Any of this has to have an economic return. And it's about truly balancing amino acids.
Very well. Stefano, any big questions that we should ask before we leave here?
Yeah, there's a question that is that Daphne posed. I don't know if Daphne wants to go ahead with that about the relationship between choline and methionine as metal donor. Basically, some says that choline can replace methionine. Is that true? What are the best practice related to choline and methionine during transition?
Yeah, I mean, the short answer is they cannot replace each other. And the long answer is if you see the pathway in the choline metabolism, so yes, methionine can get into acid-inosine methionine and it can transfer 3 methyl donors, which can synthesize ultimately phosphatidylcholine. But choline has some benefits in terms of hepatic metabolism. So I believe at the moment we have 26 experiments, and we have a very clear, and there is no question about it, that choline improves hepatic tissue metabolism while improving the productive performance. And I think among 26 experiments, 12 out of those specifically targeting the effects of choline in reducing the fat accumulation in liver. So choline was able to reduce the triglycerides from the liver, but none of the studies, at least I'm sorry if I'm mistaken, but none of the studies clearly shows that methionine was acting as a lipotropic agent. Yes, methionine has much more role in terms of memory protein synthesis and improving productive performance, but from the data point of view, I don't remember any paper where they supplemented methionine and then methionine reduced the accumulation of triglycerides in the liver. On the other hand, choline, where we conducted some very cool experiments at University of Florida, so we really induced negative energy balance in cows. So we applied the feed restriction model. So we induced fatty liver development in the cows. So cows were healthy, and we provided the feed ad libitum, and then all of a sudden we cut their feed to 50% of whatsoever they needed. So they developed fatty liver, and then we supplemented on top of that with choline, and not only University of Florida, Dr. Rick Grummer from UW-Madison, Barry Bradford from Michigan. I think there is very much clear data available that choline reduces accumulation of triglycerides in the liver, reducing fatty liver, and that's how choline improves the productive performance. Another aspect of choline is related to memory epithelial cells. So there is very cool data coming up showing that if you supplement choline during the transition period, it probably increases the memory epithelial cells. That's why even if you stop supplementing choline after transition period, the cows keep producing 2 kilograms more milk for the next 25 to 40 weeks in lactation, which tells that choline supplemented cows, they carry over effect. So yes, again, back to the point that they cannot replace each other, and both nutrients are necessary because they have several different roles to play.
Thank you, Osman. I would absolutely agree with that, and I would just add briefly that both methionine and choline are essential nutrients for the dairy cow, both the pre-calving and the post-calving dairy cow, and they both have separate needs and separate responses. And there is clear economic benefit of supplementing choline and methionine to the cow to give you performance benefits because they are essential required nutrients. And if we're going to optimize genetic potential of these cows, or the nutritional potential to achieve the genetic potential, we have got to meet their requirements for all nutrients, and that includes choline, methionine, and others.
Yeah. The other thing I'd add to that, Sean, is that, right, from a pure economic perspective, if you're just looking at methyl groups, choline has 3, methionine has 1. Choline's less expensive than methionine, so, right, cost per unit of methyl donor, choline wins, if that's what you're looking for. So, Ryan, was there a real quick question back there?
Yeah, I had one quick question. Came up over lunch, we were having a discussion. I think it's very germane to everything we were talking about, and that is, it's really for Mike. So we've got, everybody in the audience has a bunch of different products, feed additives, they're trying to utilize in their ration balancing system. From your perspective in CNCPS, how do you get those products into the library? And is it user beware and company just sends you the profile and you just put it in? Or do you scrutinize that, require research? And then the second part of that is, if you're using a commercially available program, NDS, AMTS, is there any difference between that feed library and what you have in the base CNCPS?
Wow, that's a complicated question. How much time do we have left? Yeah, I know it's getting close. I'll try to give you a quick answer. If somebody comes to us and says, we want to put the product in the library, I always ask them, what do you want? Is this, I think the hardest part about this is it a nutrient product, or does it have non-nutritive behavior? And that's the first line we draw, right? If it's a nutrient, great. Here's the chemistry you need. Here's our curation fee, right? We charge $2,500 to put a feed in the library because we have to do a whole lot of work and make sure that it's right, and we got to summarize a bunch of feed chemistry. If we're going to have to do some feed chemistry to help you, we'll charge for that, but we don't charge any more than a commercial lab because we want it in there, and we'd like to make sure if we're going to put it in there, it's correct, right? And that's for something that's nutritive. If you tell me you want to put this enzyme system in there, and this enzyme increases NDF digestibility by 12%, then I want to see every piece of data you've ever generated, and that data should include some cow studies. I don't want to see just in vitros or continuous fermenters. I want to see some cow responses. If you can't do that, I'll put it in there as a placeholder, but we're not going to do the rest of it because I've got to see some data that I know I can trust, and it has the interaction with the cow, right? About the other feed libraries, right? This is a really complex thing. So we build the reference library. It's got about 1,300 feeds in it right now. It's a lot. It's the bane of my existence relative to the CNCPS because it takes so much to manage, and we get more all the time. We're probably adding 4 to 6 feeds a month at least. Everything has to be evaluated, tested, and it's an iterative process. If you guys hand me something, you're going to give me the data, I'm going to give it back to you, I'm going to show you what it looks like, you're going to say yes or no, we're all going to agree to it, right? There are companies that won't come to us because they don't want that level of scrutiny. So they will go to the license holders, and they will ask to put their feeds in there. I always tell them, you can do that, but please don't put it in the reference library. Put it in another page, put it in another category, because I don't want you guys as users, or whoever the users are, coming back to me saying, we made a mistake, we did something wrong, that's not right, whatever. This is if you're going to put it in there, that's on you, right? There's a very fuzzy line there, because I feel a lot of times that the people see us as the policeman, right? And I don't like being in that role, but I don't want to put out false information, right? Because then that just screws the whole thing up, because we work too hard, to make sure this thing gives a pretty good answer, right? And if we can't give you that information, that's a problem.
Yeah. Thank you very much for joining us today, gentlemen. You guys been great. To our studio audience, don't get this very often. Thank you for joining us today. This has been a lot of fun. To our loyal audience out there, our loyal listeners, thank you for joining us as well. Hope you learned something. I hope you had some fun, and I hope to see you next time here at the Real Science Exchange, where it's always happy hour and you're always among friends. Cheers.